Vance Barksdale and Marcus Newell. They are senior students in the biology department, and they wanted to do an undergraduate research project. And so they built on a study that I did as a doctoral student at CU Boulder. And so I found a very interesting population that they decided that they would look at more closely. And so with that, I'll turn it over to Marcus and Vance.
All right. So I'm Marcus, and I'm here to talk to you about the DNA sequence divergence that we looked at in Texas dwarf mistletoes. Now, one thing I want you to know before we start this is it's going to be in the order of a scientific article. We're going to have an introduction, let you know what we're talking about, methods, which is everything we did, the results, everything we got out of it, and the discussion, what those results really meant to us. One thing you need to be clear of at first is what a parasite is. A parasite infects a host. So it could be something like a virus or a bacterium infecting us, us being the host, the parasite being the virus. It could be a microparasite or it could be a macroparasite. And this is an example of a macroparasite, something much larger. Nonetheless, what makes them a parasite is that they do not benefit the host. Plants can also be parasites. And much like a regular plant, plants have their rooting system in the ground to get water and their branches above for sunlight. Well, a parasitic plant, since it's living off of another plant, its rooting system, which is called the hostorium, goes into that plant and it'll extract either just water or water and the photosynthetic material that plant has made. Now, it's a hemiparasite if it just extracts water, meaning it does have functioning chloroplasts. However, if it's extracting photosynthase as well, its chloroplasts aren't functioning. It doesn't need them to be surviving in nature. Now this right here is a mistletoe. And the definition of a mistletoe is that it's an aerial parasitic plant, which is why people kiss under the mistletoe. It'd be a lot of difficulties to go on top of a tree and kiss above a mistletoe. There's a lot of different types of mistletoes. And the way they actually got their name in the first place was Mist in German means dung, and tang means branch. And when birds eat the fruit of this, they fly in the air, the seed passes through their GI tract, it germinates, they poop it out, and it lands, hopefully, on a branch where it can infect that tree. Now, that's how mistletoes typically reproduce and spread their seeds, but the dwarf mistletoe, which is the specific genus that we're looking at, they disperse their seeds by building up hydrostatic pressure in their fruit to a point where the seed just shoots out of there at about 50 miles an hour and lands hopefully on a nearby tree, and that seed can grow on that tree. It doesn't have to pass through a bird in order to live on another tree. Now, it is a hemiparasite still, so it has functioning chloroplast, but it's going to extract both water and photosynthetic materials from the plant still. It is dioecious, meaning it has male and female versions of this plant. And it infects 6 conifer trees across the world. Now, the specific type of dwarf mistletoe we're looking at is called Arcithobium divaricatum. And I'm just going to refer to this as dwarf mistletoes the rest of this presentation for your sake of not having to hear that over and over again. But one thing specific about these, they're found in the southwestern United States, and they're known to infect pinon. This is their range right here. Now, the last glacial maximum was when ice sheets were last at their peak. It's about 23,000 years ago. This resulted in plant populations that were dispersed being forced into refugial populations. So they're really limited to where they could grow when the climate changed. Now, if we're looking at a parasitic plant, they're not only limited to where they can grow, but they're also limited to where their host grows. So because of this climate change, multiple refugia formed during this last glacial maximum. We know about this because of Packrat Middens. And like your grandma, who probably has a whole bunch of stuff in her attic, a bunch of things stored up, you go up there and you look at it, it's like a time warp up there. You're like, this is from the 1930s, it looks brand new, Grandma. Did you like put it in a box? And she did, and she preserved it. Well, the Packrats do similar things, but they go gather twigs and seeds from their environment, put it in their nest, pee on it, and it preserves it. And we can go in there up to 50,000 years later, pull it out, look and see what was in during that time, and we found, well, other researchers have found remains of dwarf mistletoes in these. And once we know that there's dwarf mistletoes in them, you can carbon date the whole Packrat Midden, find out when that was. And dwarf mistletoes were, in fact, around this region of the southwestern United States during that last glacial maximum, when those ice peaks, or ice sheets were at their max. Now, Dr. Duran knew this, and so she knew that there was these 3 refugial populations here, and during the last glacial maximum. And what she was testing is to see if the current populations that we see there now arose from these prior refugees. Now, she went around, she got DNA samples, and she was looking at the chloroplast DNA of all these, and she found that there was similarities in these 3 groups, and a lot of them were similar, and it kind of correlates to the Packrat Middens, meaning that one lower down there arose from one midden, the other there, and the other there. However, there's this one star right here. I don't know which one's the later. This one right there, and it stood out from all the rest of the populations. It was more different from any of those populations than any of them were from each other, even though it was right in the middle of them. It stood out like this. This is what we saw when we looked at that. Its DNA is completely different. And she looked at the chloroplast DNA in this. She found significant difference in it, and just to give you an idea of where it was, this is the population she looked at. It has 2 neighboring populations. Their DNA was identical, this DNA was completely different. Now, to talk about methods, I'll go ahead and have Vance explain what we did.
So the method, this is kind of our experimental procedure, how we collected our data, what we did with it. And first I'm just going to give you a quick introduction to DNA. You may have heard of it as our genetic makeup. DNA is, it's like the blueprint. It says, okay, you're going to grow an arm right here. It's just how we develop is based on our DNA. And it's made up of this code. It has a DNA, you can think of it as this long chain, and like a necklace in beads. And each bead is a nucleotide. So DNA is made up of nucleotides, and there are 4 different kinds of nucleotides. And they're different on a base. They have a different base, and they're just given different letters, A, T, C, or G, and then it corresponds to a name like adenine, guanine, cytosine, and thymine. I'll just refer to them as A, T, C's, and G's. And so that you have these 4 different types of nucleotides that make up DNA. You could think of it as 4 different colors of beads, and you can arrange them to have different products. And so where you can look to find differences in DNA, because within the same species,